Good morning and welcome to the AWS Education Research Seminar Series. I'm Marcy Collinson, your session correspondent and the Scientific Research Programs Manager for the AWS Research Team. Today we are delighted to have Dr. Orly Alter presenting on multi-tensor decompositions for personalized cancer diagnostics, prognostics, and therapeutics. Orly is a Utah Science, Technology, and Research, or USTAR, Associate Professor of Bioengineering and Human Genetics at the Scientific Computing and Imaging Institute and the Huntsman Cancer Institute at the University of Utah. She is also the Principal Investigator of a National Cancer Institute Physical Sciences and Oncology U01 project grant. Orly is the inventor of the eigengene. She pioneered the matrix and tensor modeling of large-scale molecular biological data, which, as she demonstrated, can correctly predict previously unknown physical, cellular, and evolutionary mechanisms. She received her PhD in applied physics at Stanford University and her MSc magna cum laude in physics at Tel Aviv University. Her PhD thesis on quantum measurement of a single system, which was published by Wiley Inner Science as a book, is recognized today as crucial to the field of gravitational wave detection. We will reserve the last 5 to 10 minutes of the session for a Q&A portion, but please feel free to place any questions that you may have during the presentation in the questions queue in the GoToWebinar toolbar, and we will read them aloud later on. So without further ado, I'll pass the microphone over to Orly. Orly, please take it away.
Thank you, Marcy. Oh man, sorry everybody. Hi everyone, it's great to be here. And this is Orly Alter from Salt Lake City, and it's great to join with Marcy and with all of you. As Marcy was mentioning, I will talk about multi-tensor decompositions for personalized cancer diagnostics, prognostics, and therapeutics. And I just wanted to mention that we have a startup, eigengene, to bring our mathematical technology to the clinic. Oops, sorry. So, cancer clinics and hospitals, and really clinics and hospitals in general, are now adopting high-through biotechnologies. These biotechnologies came around the year 2000, I would want to say, and with the Human Genome Project, and they enabled for the first time to measure the complete genomic signals that cells generate and read as they go about cellular processes. So, for example, we have here the flow of genomic information in the cell, where DNA gets transcribed into RNA, gets translated into proteins. And what these high-throughput biotechnologies enable is measuring DNA abundance level of all the genomic loci. For example, the human genome has 3 billion nucleotides, RNA abundance levels of, say, all the genes, and for example, the human genome is estimated today to have 20,000 genes, and the binding of proteins to DNA and to RNA. And this brings the question of what to do with this data. The question existed before when the technologies came about, but now that they're being adopted by hospitals and clinics, the question becomes even more acute. How do we understand what the data tell us, and how do we get clinically actionable information from the data? So, coming from physics, where theory is built upon data and then informs experiments to acquire additional data, we decided to start with the singular value decomposition. The singular value decomposition underlies theoretical physics. But for example, the physical activity of this prism that takes white light and separates it into its color components is mathematically described by the singular value decomposition. Applying the singular value decomposition to genomic signals, for example, this 2-dimensional table or matrix of genomic data, we were able to show that the singular value decomposition similarly, in molecular biology, uncovers from the signal or separates the signal into the cellular processes and the cellular states, the corresponding cellular states that give rise to the data, that compose the data. I should also mention that some of what composes the data are batch effects, and we will definitely talk about that as well. But, oh, and I also wanted to mention that by using the singular value decomposition, this is where we invented the eigengene. If we have here this data set where in this example, we have along the x-axis samples from yeast during cell division at different time points during cell division. And along the y-axis, we have all the yeast genes and tabulated our abundance levels of RNA or gene expression in yeast during cell division. So this data set, the right-hand singular vectors that the SVD gives us, the patterns that correspond to the cellular processes, we call them eigengenes. This is where the eigengene comes from. But very quickly on, we realized that data don't always come in the format of a single two-dimensional table or a mathematical matrix, if you want. And really, the data could come in the form of multiple tables. So for example, here we have two-dimensional tables or matrices. And we want to put these different types of data together. In this particular example, we have one of the matrices is similar really to the cell division RNA expression from yeast that we have here in the SVD example. But now we're adding the binding of proteins to DNA and specifically transcription factors and replication initiation proteins that are known to drive the RNA expression. So how do we put this data together? Well, we generalize the singular value decomposition to, or use a generalization of the singular value decomposition, specifically tailored as a data science tool here to enable us to correlate the two data sets. And by doing that, we might find correlations that correspond to causal coordination. We actually seen some of those that were known, but then we found some that were previously unknown, and we could predict, which is what we did, new mechanisms. So, you know, again, coming from physics course, so correlation could be causal coordination, but not necessarily so. Predicting a mechanism is great, but we need to test it experimentally. And we tested experimentally our prediction with John Diffley of Cancer Research UK. The test essentially verified our predicted mechanism, which was not known before. And in order to test the prediction, we needed now to collect data, which is what we did with John, data similar to the data I showed you earlier, where along the x-axis we have time points during cell division in yeast. Along the y-axis, we have all of the yeast genes and tabulated our RNA expression levels in these yeast cells at these different time points, and but under different conditions. So we have a wild type like we had before, but we have two additionally treated conditions in order to see that really we have here a causal relationship. These data are not a single two-dimensional table anymore. They're also not two-dimensional tables. Really, they are a single higher order cuboid, if you want, of data, because we have here this additional access, the additional access of the conditions under which we collected the data. And in order to interpret the data, we used another generalization of the singular value decomposition, which again, we formulated as a data science tool. But just to let you know, we were excited, obviously, to conclude that this kind of matrix and tensor modeling of molecular biological data can correctly predict previously unknown mechanisms. To find patterns in this structure, this cuboid or mathematically it's a tensor, a higher order structure of data, we generalize the singular value decomposition by using a formulation described here by Levin Delathawer, where we are still going to find patterns along the genes, patterns along the time points, and patterns along the conditions corresponding to the axis of the data we have here. These patterns are still going to be orthogonal to one another, but the core tensor here that we have in the data is not going to maintain the same mathematical properties as in this SVD. In the SVD, you might remember the core is a non-negative diagonal matrix. Obviously, here we have a tensor. The tensor is not necessarily non-negative, and it's definitely not necessarily all orthogonal. So this is, I wanted to point out, a big field right now for research in terms of generalizing the SVD from a two-dimensional matrix to higher order tensors. We cannot keep all the mathematical properties of the SVD by using just one mathematical formalism, so we need to choose. This is the formalism that we chose, but in choosing it, we also needed to understand how to interpret the patterns we find in the data. So now we're finding patterns, as I said, across the x-axis and the y-axis and this additional z-axis, and these patterns have a coefficient or magnitude in the data that comes from the core tensor, and we realize that what we want to interpret are those rank 1, if you want, outer products of these vectors. So we want to interpret the combination of the patterns, and we want to interpret them based on the coefficients in which they appear in the data in a decreasing order. We also had to define entropy. We had to define degenerate subspace, which are things that we had before in matrix algebra, but not necessarily in tensor algebra and not necessarily for the tensor SVD. And I also wanted to point out, I mentioned batch effects. So batch effects is, all of us who work with genomic data are aware they are part of the data, but they're not just a part of genomic data. So early on, I learned from Sabine van Hoffel that batch effects actually affect every possible high throughput acquisition of data. So we see them in sky surveys, in astrophysics, we see them in biomedical signal processing, traditional biomedical signal processing, for example, looking at EEG and ECG and MRIs. And in all these cases, the batch effects might manifest themselves as baselines that are superimposed on the data, and they differ between technicians, they differ between dates, they differ between hospitals. And in those cases, there are already computational platforms implemented in hospitals, which were designed by medical physicists for the most part. And the underlying mathematical framework there to separate the batch effects from the biological signal, biomedical signal of interest, is the singular value decomposition. So here we have in our data, for example, just an example of how we see those batch effects separated in this cell cycle time course. We have here again the time points across the time points during cell division in yeast, and those are the even time points, those are the odd time points, and you could see that while we have some patterns that look periodic and there is a constant pattern, there is also a significant pattern in the data that is essentially a step function between the even time points and the odd time points. And I should mention that we received the yeast samples to run on microarrays in my lab. We received them in separate FedEx boxes from John, so we kept the batch effects separated between the even time points and the odd time points, and here we see them. We see that pattern coming through. We also kept it for the hybridization and for the experimentalist who ran the experiments, and so we kept all these batch effects coordinated so that we will be able to easily tease them out of the data. And part of the easiness is really thanks to the singular value decomposition. I should also mention, I think I might have forgotten to mention it earlier, that here the data, so the data, the numbers that are tabulated, negative numbers are tabulated in green, and they represent, say, in this particular case, that it's gene expression below steady state, and positive numbers are represented in red, and they represent, in this case, gene expression levels that are above steady state. So we were excited about being able to find a correlation that suggested causal coordination, and that reminded us of several instances in physics when this happened. And it wasn't just quantum mechanics, even before in classical mechanics, and even before that, when Kepler was looking at Brahe's data. And in this case, Kepler had a table, one two-dimensional table of very high quality data. So experimentalists out there, the quality of the data matters, even if we're able to separate batch effects from the biology in the data. And the table that Kepler had, it has, let's see, along the x-axis, these are celestial objects, if you want. So we have here planets and moons, and along the y-axis, these are time points, and tabulated are positions in the sky of those objects at the different time points. Kepler found patterns in the data, and they corresponded to his 3 laws of planetary motion. The first law of planetary motion corresponded to an ellipse he found in his data, and while arguably the patterns we found in the cell division experiments that I showed you earlier, those patterns might correspond to ellipses. Normally people don't find ellipses in their data. Kepler was able to find these ellipses easily because he had the mathematical technology of conical cuts. He has the mathematics to enable him to tease ellipses that were there out of the data and convince himself that, yes, these are really the patterns that he sees. Kepler's third law of planetary motion, that was the law of correlation. He never visualized it. He didn't have the visual approach to visualize it, even though today, say, if you look at NASA's website, this third law is described as a law of regression correlation. He didn't have the mathematical technology to compute very robustly this regression correlation. Today, we might use the singular value decomposition and total least squares and really the pseudo-inverse projection to calculate this kind of regression correlation and get at Kepler's third law, but he didn't have pseudo-inverse projection, he didn't have the singular value decomposition, and he didn't have total least squares. So it took him about 9 years to convince himself of this, of his third law. Going from the first to the third law, the correlation was the most difficult thing for him to find and convince himself of. And his third law is the law that ultimately gave rise to Newton's law of universal gravitation. So it was obviously very important in physics and in science in general. And the reason I'm telling you this is to give you an example of how having the correct mathematical technology matters, and also to let you know that finding correlations, we shouldn't be sneezing at our ability to find correlations in the data. So then we asked ourselves, well, now that those technologies are adopted by hospitals and clinics, what is the mathematical technology that might be missing? What is the mathematical technology essentially for personalized medicine? And so in personalized medicine, I will start by showing you this example. We started with it as a toy example, but where it's actually very close to getting to the clinic, which we're excited about. In personalized medicine, the structure of the data is a little different than what we've seen so far. Here we have, say, tables, two-dimensional tables. For example, in this example, we have here one two-dimensional table on the left and a second two-dimensional table here on the right. And these tables are, they share the same x-axis, or they have a mapping in their x-axis. Essentially, those are the patients. It's the same patients in both data sets. And we measure different things about the patients. Obviously, we can measure more than two things, but as I said, for the toy example, let's look at just two things. But this is the structure of data in personalized medicine. We have multiple matrices, the same x-axis, very possibly completely different y-axis, and different types of data tabulated in them. And then how would we find correlations in this kind of data? How would we find patterns in these kind of data? How would we generalize the SVD to this kind of data? So there existed the generalized singular value decomposition, which was proposed by Charlie Van Loen of Cornell, and then was given robust computation algorithm by Michael Saunders from Stanford and Chris Page, his collaborator. And so we use that we use that as our beginning, and then we formulated it to be a data science tool for us. This is how the formulation works. We have these two data sets. Just like the singular value decomposition would separate each of them into three matrices, we are now separating the two data sets into each of them into three matrices. They have two left-hand singular vectors. We have these two matrices that capture the left-hand singular vectors for both matrices, one for each. We have two core matrices that are still non-negative and diagonal, but they're different. They tabulate different numbers along the diagonal. And we have a single set of right-hand singular vectors that capture patterns of variation across the shared axis, essentially across the patient. So the mathematics tells us, well, we need all these patterns in order to describe the two data sets. But these patterns can come with different significance in the two data sets, and the difference in the significance we decided will be based upon the coefficient or the magnitude of these patterns in the core non-negative diagonal matrices. So just to tell you what these data are, finally, these data, they're data from the Cancer Genome Atlas, which is a fantastic initiative of the National Cancer Institute, where they collect omic data, many kinds of omic data, and actually additional data like imaging data from different types of cancers, from cohorts of patients. The data are publicly available. And in this case, we're looking at DNA copy numbers or a genomic profile of the normal blood samples from a set of patients. And here we have DNA copy numbers or genomic profiles of the tumor samples of the same patients, with one-to-one matching between the patients. The patients are patients of astrocytoma. Here we're looking at astrocytomas grades 2 and 3, as well as astrocytoma grade 4, and some of you might know it, the grade 4, which is the most devastating grade of the disease, is glioblastoma or GBM. And when we're looking at these data, if you want our toy example, again, black means steady state. We see that even in the normal data set, there is variation across the patients. We see here, this is the X chromosome. Here we have all the autosomes, and here we have the X chromosome. And we see along the X chromosome that some patients appear to have a deletion of the X chromosome. This is not completely surprising. This is normal variation between the patients, and specifically the variation between males and females. We see this variation also manifested in the tumor data set, but obviously in the tumor data set we have additional variation. There are gross changes in the genomes of these of the patients. The tumor genomes are very different from the normal genome. They include amplifications of whole chromosome arms, say chromosome 7, deletion of amplifications of all chromosomes, say chromosome 7, deletions of chromosomes and chromosome arms, say chromosome 10, and the short arm of chromosome 9. So we see gross variations combined that are maybe tumor specific, combined with variations that come from the normal genomes. And what the GSVD enables us to do is essentially separate what is specific or exclusive to the tumor from what is exclusive to the normal and what is common between the two data sets. It's like the prism, but it's a comparative prism, if you will. And essentially, those patterns here across the patients that have coefficients which are similar, those patterns will correspond to patterns of variation that are common to the tumor and the normal, and those patterns that have coefficients that are very large in the tumor, very small in the normal, those would be exclusive to the tumor, and vice versa. This is what the mathematics tells us. So what do we find in the biology? Yeah, these slides are just to mention that, as I said, we needed to add additional formulations in order to take this computation and make it a data science tool. So what did we find? We found a pattern that is tumor exclusive in this particular data. These data that I showed you, they were measured with whole genome sequencing. The 3 billion nucleotides that we have in the human genome, we beamed into about 3 million beans here, really just for the computation. And so this is the pattern that was tumor exclusive. You see all these aberrations in the genome of the tumors. And this pattern reminded us patterns that we found before, specifically when we looked at, we ran a similar exercise looking not at whole genome sequencing of astrocytoma patients in general, but looking at just Affymetrix microarray data measured from the lower grade astrocytomas, or looking at the Agilent microarray data measured from the glioblastoma, the grade 4 astrocytomas. So we find very similar patterns, and in all these cases, just like here, The pattern of variation across the patients, you see this data set, it's now, it's rotated 90 degrees. These are the genomic coordinates. Here is the X chromosome, and here are our patients. So the pattern of variation across the patients, as the mathematics says, is exclusive to the tumor, separates the patients based upon survival. And the separation is striking. For example, this group of patients here, they have median survival time of about 1 year. This group of patients here, they have median survival time of about 5 years. And if we were, this is because we're looking at the astrocytoma in general, if we were looking at just glioblastoma, we will have a variation of 1 year relative to 3 years, still very statistically significant. And I want to point out that it is striking that the mathematics finds the same pattern, even though we're looking here at different technologies. Normally, different technologies allow assessing copy number variation with of any one genomic locus at less than 70% reproducibility. Here we repeat the whole exercise on completely different data sets, and we find this relationship genotype-phenotype relationship very reproducible. The relationship had some known biology. It suggests some previously unknown biology, including some potential drug targets. So I won't go into these details, but please ask me if you're interested. But then the question is, what are the other wavelengths that the GSVD, the comparative prism, finds in the data? What are the other patterns that we separate from this tumor-exclusive genotype-phenotype relationship? Well, there are some tumor-exclusive patterns that correspond to batch effects, specifically effects that are known to affect whole genome sequencing. And this is the GC content for those of you who know about the kind of batch effects that are frequent in whole genome sequencing. And in this particular case, they seem to vary between genomic characterization centers. We see normal exclusive patterns, and again, they separate batch effects. The batch effects in this case, they vary between the tissue source site of TCGA. Previously, we saw similarly separation of batch effects for microarray data. And just so you know, every technology has completely different batch effects. I wonder, so this pattern, oh, okay. So this pattern here that we have for the whole genome sequencing, it looks very different from this pattern here that we have for the adjuvant microarray data, for example. Oh no, sorry, that one was for the Affymetrix microarray data. These are the adjuvant microarray data. Anyways, we see all these batch effects, and they vary between tumor hybridization plates, tumor sample centers, and all of those contributing experimental variations that essentially it's impossible to create the data without them. And we don't need to worry about modeling these effects, or we don't need to worry about figuring out that they are there, because the GSVD separates them into a different wavelength, if you want, just like our prism in the beginning. And what are common patterns between the tumor and the normal? Like I was alluding to earlier, the most striking common pattern that we find, it's a pattern of variation across the patient, reproduced here twice. These are the tumor data, these are the normal data, and we see in the normal data that this pattern very clearly appears to be some kind of noise around steady state for all of the autosomal chromosomes 1 through 22, and a deletion of the X chromosome. So we have a deletion of the X chromosome relative to the autosome in some of the patients. And indeed, these patients we find are the male patients, And these patients here that don't have the deletion, they're the female patients. And this pattern is common between the normal and the tumor because a tumor of a male patient starts from a normal cells that have one less copy of the X chromosome than a tumor of the female patients. So this variation in the X chromosome to zeroth order is not necessarily contributing here to the disease. It's separated as a normal variation that is common to both data sets. And again, there are many normal variations that we don't actually know about when we look at the data. So we don't have to guess what these variations are. The mathematics just tells us what they are and separates them into different wavelengths. And this is especially important when people end up, which is the common approach today, people end up throwing the X chromosome out of the analysis because this is considered a confounding factor. They end up throwing the Y chromosome data out of the analysis. They end up throwing the mitochondrial DNA, so much data gets thrown out where we know that we can expect normal variation, but then there are all those instances of normal variation that we don't even know about, and we cannot throw it in advance, and this is still confounding all these particular analyses that cannot separate common variations into a different wavelength like we do here with the GSVD. I should also mention that all of us who work with data know that databases come with data labels or attributes that have some errors in them. This is just, part of what we work with, not just batch effects, but also labels are incorrect. And essentially by making this classification with the GSVD, we were able to correct the labels of TCGA. Some of those patients here that seem like males, they were labeled as female and vice versa. You never know because it's biology, and we contacted them and they said, oh, well, thank you for helping us correct our database. So it's nice to have mathematics that helps you pinpoint these kind of possible errors when they arise, so you could go back and check what is actually going on there. And regarding the difficulty of throwing out the X chromosome from these analyses, or the Y, or the mitochondrial DNA, I wanted to point out that this is such a common pipeline that actually the Cancer Genome Atlas, they had a paper on ovarian cancer that was published in Nature. And the first thing that the pipeline does is throw out the X chromosome. So this gynecological disease, the X chromosome data never made it into the paper. The situation is such that, those of us who are interested in analyzing data from the X chromosome, here's a notice for funding from the National Human Genome Research Institute, because everybody tends to throw out those data, but not us. So I mentioned that we can find some new drug targets with this analysis, and specifically here, unlike maybe other analysis where we find potential drug targets, the drug targets correlate with patient outcome. Normally, it seems when people decide on testing a drug, they don't so much look for potential correlation with patient outcome. They look more at, well, is this drug targeting a gene that is in some kind of a pathway that might be relevant for cancer, like the cell cycle? Well, here, not only we find genes that sit in those pathways, and these pathways include very many genes, mind you, but we also can say which ones the mathematics predict on their own to correlate with outcome, and therefore maybe targeting them might prove beneficial for the patient. So this pattern of variation across the patients, it essentially classifies the patients, or the classification of the patients based upon the correlation with the genomic pattern that we found. We realized after we found them that they give us a classification that is statistically better than and independent of the best other indicators, which for astrocytoma in general included grade, and for glioblastoma alone was only age. And it is better than the recent indicators that look at single changes in a single genomic locus, for example, MGMT promoter methylation or IDH1 mutation for glioblastoma. The accuracy or the concordance index in this analysis was 95%, and but I should tell you that our range in all the different analyses we ran is in between 75% to 95%. So this one is a particularly, I guess, successful example. Anyways, the fact that we're statistically better is great, but we're also statistically independent of all of the other indicators. And that says that this information that the genomic pattern that the GSVD found for us in this data is actually not being used to make clinical decisions. It's not affecting anything that's used to make clinical decisions. So we concluded that the GSVD underlies the mathematically universal description of the genotype-phenotype relationships, and I want to emphasize that other machine learning methods miss. Really what we think is that we have here a theory. You may not necessarily agree, but you must agree that other methods missed this. Well, they missed it so far. The data are publicly available. So many people tried so very many different algorithms on the data to try to tease out anything that might correlate with the patient outcome or anything that might be meaningful. And we found here a relationship that is meaningful for adult astrocytoma at large and is suitable for all genomic profiling technologies. I don't want to go into all the details. I want to emphasize that our ability to do that, and especially to use the whole genome sequencing data that we used here, is really thanks to our moving to the Amazon Web Services or AWS platform, because the data, when you look at whole genome sequencing, they really start to grow in size. We had about 20 terabytes just for this example of only 85 patients. So we have 20 terabytes of data, and we're downloading now data that are closer to the petabyte scale to use. And at the same time, we have very small cohorts of patients. And again, I want to emphasize this because so many algorithms today, specifically convolutional neural networks, they find fantastic results looking at imaging data where you have, say, million images and they're all, well, hopefully perfectly classified. But here we only have 85 patients. And the number of beams that we looked at, the number of, so that's the number of columns in each data set, the number of rows, it was 3 million in this particular instance. So the GSVD is a robust mathematics to do something that other methods cannot do, and that you need high performance computing in order to do it. I also wanted to mention that just like in our prediction of a mechanism, which we then went and tested it experimentally, here again, to have sort of an experimental test of our predictor, we ran a retrospective clinical trial. We did this with Jill Barnholt Sloan of Case Western Reserve University, and we were able to validate our results. This is under review, so I won't show you the images from the publication. I will just tell you that yes, we validated that the pattern predicts survival better than age. And the patient age at diagnosis, which as we all agree is not the most current technology, right? it has been the best indicator of the disease since 1950. And it's not because people did not try to associate these kind of genotypes and copy number alterations with the patient phenotypes. They tried. There were so many attempts to try to do this, not just looking at TCGA data, but even before. And they were all unsuccessful. They all will tell you, well, we find all these genotypes and we don't know, we're unable to correlate them with patient phenotypes. So these alterations were observed in the data before, but were never able to be brought to the clinic in any way or to even inform the understanding of the biology of the disease. So this is the proof of principle that the GSVD is uniquely suitable for discovering clinically actionable genotype-phenotype relationships, and the relationships are clinically actionable both in terms of the prognostics, the diagnostics, and the therapeutics of the disease. And I see that I'm running out of time, so I will not go into much details. Please ask me. I wanted to give a shout out to our high performance computing platform of AWS, which essentially facilitated this work. As I mentioned, the sizes of the raw data that we use, and we like to use raw data actually, it separates better batch effects. The sizes of those raw data are large already and growing. The number of computations that we need to perform is large and growing, and specifically because we always test the robustness of anything that we get to perturbation of the data. So that means we introduce hundreds or thousands of perturbations to the data sets, and we want to see that we get the same biological results, at least qualitatively, if not quantitatively, looking at these different pre-processing steps of the data, even if we use very few pre-processing steps. And really, it's using the AWS platform that enabled us today to classify the GBM patients based upon this pattern that we found in the data with greater than 99% reproducibility. And I mentioned earlier that experimental batch effects limit reproducibility when we're looking at just any one genomic locus to less than 70%. There's still a lot more work to do, experimental work, and I want to give here a shout out to the Physical Sciences and Oncology Network that I'm a part of, and please go and check all of my colleagues' fantastic projects there. There's so many characterizations that they bring to the table that we would like to deploy here. There are so many additional models that we're interested in constructing, and we started to do that looking at cancers beyond astrocytoma. I should mention, if I run out of time, we have results also for adenocarcinomas, lung, ovarian, and uterine. There are disorders beyond cancer we could look at. There are omics beyond DNA copy numbers. There are so many additional data that we can model. There is a lot of work for mathematicians, including coming up with computing and distributed computing algorithms, which then can be deployed at the AWS platform. But just to tell you about the direction of my lab, so I mentioned we're looking at additional cancers. We're also looking mathematically, just like we did before, at generalizing now the GSVD to more than two-dimensional matrices, say to multiple two-dimensional matrices. This work is with Sri Priyaponapalli and Michael Saunders and Charlie Van Looyen, who I mentioned before. Priya, she's now a principal investigator, a principal researcher principal scientist and a senior manager at AWS. So we're very proud of her. And actually, she introduced me to the platform before she started at AWS. And so we could look at multiple data sets. We could look at data sets that have more than 2 dimensions and multiples of them. We can, again, we need to define the mathematics that we're looking at, the work with Priya, the algorithm that I developed with her. We started with Jean Golub of Stanford University, and we continued with Michael Saunders and Charlie Van Loen. We needed to prove many theorems in order to take this computation and make it a data science tool. So for example, proving this eigenvalue inequality took us 5 years. And actually, I should mention the experiments with John Diffley of Cancer Research UK that I showed you earlier. They also took us 5 years. So things take time. Using these kind of mathematics, as I mentioned, enabled us to find patterns in adenocarcinomas. And just like before, the patterns were not found by other algorithms, but are consistently found by the GSVD and its tensor generalization in different types of platforms, in data measured in different types of platforms. There are some biological pathways we can suggest underlying this. There is a pattern in the X chromosome that actually is here involved with ovarian cancer patients' outcome. There is separation of batch effect. We're again statistically independent of the best other indicator. Which means that we are finding information that's not being used in the clinic, and we want to bring it to the clinic. Here are additional generalizations that we can think about. And so this is really taking all the mathematics I showed you so far and going from 1 2-dimensional data set to multiple higher dimensional data sets. This work is also in progress. I wanted to mention that additional way to find principles of nature is by finding the statistics that underlie the data set, and the GSVD, the singular value decomposition alone already enables us to do that. But I won't go into the details here as well because I'm running out of time. I also wanted to mention that why are we doing all of this? So yes, we want to bring our mathematics to the clinic, and we want to be able to one day have mathematics as effective in biology as it is in physics today. Please tune in to our session. We have an educational session, an invited educational session at the annual meeting of the American Association for Cancer Research. And I will be chairing it and presenting there together with Rick Stevens of Argonne National Laboratory. And of course, thank you to all of my fantastic collaborators and mentors. And specifically here, I really want to thank Jill Barnholt-Sloan. We wouldn't be able to run our retrospective clinical trial without her help. She runs the tissue source site for the Cancer Genome Atlas at Case Western Reserve University. I also very much want to thank my lab alumna, Sri Priya Ponapali. As I said, Priya not only did fantastic work developing algorithms when she was a graduate student in my lab, she also introduced me to the AWS platform. And of course, I want to thank my funding from the National Cancer Institute, from the Physical Sciences and Oncology Network. And thank
Orly, Yeah, great. Sorry, I hate to interrupt. I know this has been a fantastic presentation, but we are at the portion of for the Q&A, and we do have some questions that I'd like to ask you. So one question that came through, not related to Orly, is just in general, is this webinar recorded? And the answer is absolutely yes, it is. And you can find all past presenters, presentations, and recordings on the homepage in the Past Presenters tab. So hopefully that answers that one. Now I'm going to go ahead and jump into the ones for Orly, and I'll just read them aloud. Could you discuss how other clinical data, i.e. not genetic, may complement or be integrated into your approach to further individualize care?
Oh, that's a fantastic question. And yes, so our algorithms are completely non-domain specific. That means that those data slabs that I showed you earlier, if I can find one to scroll to, those data slabs don't necessarily have to be genomic data. They could be any kind of data. So for example, if I'm scrolling quickly to this GSVD example, the toy example we started with, I guess it's not so quickly, but anyway, each one of these matrices that we're integrating together, each one of these matrices could be any kind of data that we measure in this particular instance about the same set of patients, right? So we have here the same set of patients in these two data sets. Maybe I should have brought you to the slide on Priya's algorithms. We have here multiple data sets. We have now algorithms for multiple data sets from the same set of patients. And by vectorizing the data, so here these are genomic data that we vectorized, but you could easily imagine taking MRI data and vectorizing it, or taking any kind of other data, maybe data that are measured by digital healthcare wearables, or really any data that one can envision that might be of any relevance, and we just put them in their own two-dimensional matrix, and then the mathematics is going to find for us what is common between the different data sets, or between subsets of the different data sets, because we'll have more than two, and what is exclusive to either one of the data sets.
Awesome. Okay, I'm going to move on just because we're running out of time. So the next question is, just to clarify, could it be said that in essence, your method is looking for a principal component that best separates responders from non-responders?
No, actually, this is completely different on several levels. First, we don't include the label of response in our analysis. I'm trying to scroll into that slide I have in the end as well, but we don't include the labels in the analysis. In the analysis I showed today, we only include the genomic data. So the correlation with the labels of the patients, this is something that we find when we're interpreting the patterns that we find in the data. These algorithms, they're discovery algorithms. They are not learning algorithms, which also makes them less sensitive to labels that might be incorrect, I guess, as I mentioned earlier with the case of gender. So this is absolutely different from a learning algorithm. This is a discovery algorithm. We don't incorporate this information in the analysis, the information of response in the analysis. It's actually beautiful to us that the mathematical question alone of what is exclusive to the tumor genome relative to the normal genome finds out a difference between the patients that can be that is interpretable in terms of the patient phenotype, because the patient phenotype is never coming into the equation. So this is one important difference. Another important difference is that the SVD is actually different than principal component analysis, even though it is used for its stable computation. For the sake of time, I won't go into all the details, but yes, so the building block we're generalizing is different from principal component analysis, and the the approach we're using, the data we're integrating in order to find the patterns completely doesn't include the labels.
Okay, great. And we did have a couple of other questions, but unfortunately we're out of time. So Orly, I don't know if you want to just share your contact information with everybody and they could maybe reach out to you individually.
Sure, I'd be delighted to. I think I had it on my first slide, to which I'm frantically scrolling back.
Okay, no, that's fine. Don't worry about it. So for those of you who still have questions for Orly, we will be posting her presentation deck along with the recording of this session on our webpage. So you can feel free to find her contact information there and reach out to her if you have additional questions. I'd like to take a moment just to thank you, Orly, for all of your time and your wisdom. This was a fantastic presentation, and we hope that you had as much fun as we did with putting it together. So thank you, thank you, thank you. I also wanted to just tell our audience that we will have our February research seminar series session coming up, and that will be with Dr. Helio Costa, who is the instructor of pathology at Stanford University and a geneticist in molecular pathology within the departments of pathology and biomedical data science at Stanford Medical School. And he will be discussing precision medicine at scale, developing a global open source platform for interpreting the clinical genome. And with that, I hope everybody has a fantastic rest of your week. We appreciate your time and your participation, and we look forward to seeing you again on the next session we have for the AWS Education Research Seminar Series. Thank you.
Thank you. Bye.